knowing as much of this normal anatomy as possible, and they will review some of it, but not all of it. So it will help you. There's really nothing much we can cut out. We've got to introduce you to all the little nooks and crannies and different parts of the eye. So bear with us and realize that it will pay off when you have your ophthalmology course, or it'll even more pay off if you decide to become a board certified ophthalmologist. Just be sure you have a taste for emergencies, because ophthalmologic, that's a mouthful, cases are usually an emergency. You'll get a lot of emergency cases. That's what I'm trying to say. Okay, so we're going to start with the fibrous tunic. What I'm going to do is I've got the handy dandy lecture notes over here, and I'm just going to use that to guide me on what to go through and what to talk about, so that you can use that as a reference. So we looked at the fibrous tunic yesterday. Gosh, was it only yesterday? And we talked about the sclera, the cornea, and the limbus, where the two connect. So that is your outermost layer, and that is called the fibrous layer because it's made up of fibroblasts or fibrocytes and a lot of collagen. So now we need to look a little closer. We won't look any closer at sclera. That's just dense irregular connective tissue, but the cornea has some layers that we need to take a look at. Okay, so let's zoom in on it. Now remember our directional terms, so we'll be anterior here and posterior here. Okay, so anterior is going to be towards the, well, anterior surface of the eye, what you would touch if you could touch your cornea.
Very much so impressed.
It's usually very
because sometimes she gets stuck on COVID floors whenever there's, you know, since she's the most experienced cardiology nurse on the floor, whenever I'm a patient.
This anterior epithelium, would you expect it to be cornified or non-cornified? I hope you're thinking non-cornified, because that would be correct. If it became cornified, it would be hard for light to pass through it. It does become cornified during pathology, but it should not be in a healthy animal. So this is called the anterior epithelium, stratified squamous, non-cornified. Now it has a basement membrane, and that is called the subepithelial basement membrane. Okay, so there's no surprise in that. Then we have the corneal stroma. And this is really unusual. It is made up of fibroblasts. You can see them, their nuclei here, and ophthalmologists will call them keratinocytes. And their collagen layers are arranged in alternating perpendicular layers. So here is a schematic. Here is your epithelium, your basement membrane, which some ophthalmologists will call the, excuse me, the Bowman's layer. Then we have our lamellae, or our alternating layers of collagen fibers. So these are running in this direction, and then the next layer in this direction, and then it repeats, and then it repeats, and then it repeats. So how does that look on the slide? Well, you'll kind of see what might be some cross sections of collagen fibers that would be coming out of the field of view right toward us. And then you'll kind of see some waviness where the collagen fibers are going in this direction. Okay,
you know,
so alternating layers.
Charge of that floor.
And believe it or not, that alternating layered design,
but she gets tested regular,
to pass
and she's still negative.
All of these collagen fibers. So of course, this is all here in real life. So this is insane. So
there you go, Ghostbusters.
Stroma is what we call that, or potential and it has another basement membrane. Something strange. So here's the posterior epithelium. This is a really low symphysoidal epithelium. And it's called by ophthalmologists the endothelium. That makes us pathologists find our endothelium by classical definition. But they call it that because it is continuous with what is called the trabecular meshwork here in the iridial corneal angle, and that is continuous with blood vessels that drain the aqueous humor. Oh, yeah. So they call it an endothelium, and we can't get them to stop saying that, so we have to tell you that.
The only way to survive 3 hours of histology.
Now, this basement membrane between the stroma and that posterior epithelium is really thick, and they have another name for that. They call it decimated membrane. We have two membranes, an anterior subendothelial basement membrane, and
oh, that's great.
basement membrane called the posterior, excuse me, limiting membrane or Descemet's membrane. But those are both basement membranes. And in this schematic, no, I got rid of the one that had a kind of a 3D view of all these layers. All right, so
way to survive 3 hours.
Those layers are pretty easy. They classically show up on the exam questions. So, you know, and clinically, boy, those layers are really important to you, especially the endothelium back here. If this endothelium doesn't remain intact, then the cornea will get opaque. But you'll learn all about that in your ophthalmology course. And of course, we look at the limit, which is that junction between the cornea and the sclera. You have your somewhat regularly arranged, specially arranged fibroblasts and collagen fibers in the cornea, and then you just have your regular connective tissue of the sclera.
I think we just got Zoom bombed.
Then the next in line in your notes is the vascular layer. And remember, that was the choroid, the ciliary body, and the iris. So we're going to run back here to the choroid, and we're going to take a look at it. Now, the choroid is associated with the retina. So it's between the visual retina and the sclera. So here we are on the posterior surface of the eye right here. And this is the sclera. Here are some extraocular muscles. This is artifactual separation. Here is the choroid, and then here is the visual retina right here. Now, the choroid has 5 layers, potentially has 5 layers. One of them, the first layer, I think is kind of silly. Now, I don't know why we name it that or why it has a name, but it does, and we have to tell you about it. It's called the suprachoroid layer. And it's really where this artifactual space occurs, where this artifactual separation between the choroid and the sclera, that is that suprachoroid layer. I have no idea why they named it. Rather silly, but they did. Okay, next is the vascular layer, and it's the bulk of the choroid. So it's all of this collagen with these larger blood vessels in it. That's the vascular layer. In some species, these vessels are very thick, especially the arteries. They have thick walls in some species. And in some species, there's a heck of a lot of melanin in this area. Now, this is a pig, and he doesn't have much melanin right here. Okay, but we'll look at some other slides, and you'll be blown away by how much melanin is in this area. Okay, so that's the vascular layer. The next would be the tapetum lucidum. If there is a tapetum lucidum, which there isn't in the pig. So the pigs don't have a tapetum lucidum, but we'll look at some examples of a tapetum. Next is the choriocapillary layer. And this is just a capillary bed that is right up against the pigmented epithelium. The retinal pigmented epithelium is this layer right here. And I'm going to go real slow, and I'm just going to kind of zigzag. So from here to here, you can see a nucleus here, a nucleus here, a nucleus here. It kind of looks like a simple cuboidal epithelium, and that's actually part of the retina, and we'll talk about it later. There's a little basement membrane right here, and that's called the basal complex. That basement membrane is shared between the capillary bed and the retinal pigment epithelium. Okay, so that's the basal complex. That's another layer. This choriocapillary layer is just the capillary bed of the choroid, hence the name choriocapillary layer. So what about this tapetum thing? Well, what does it do? Well, it reflects light. It gives species that have that tapetum a second chance to excite the photoreceptor pigments in the photoreceptor cells of the retina. Now, this is a carnivore, and carnivores have what is called a cellular tapetum. So there are 2 types of tapeta. One is cellular and one is fibrous. And this is a carnivore. Now, let me orient you. This is the sclera. Then when you start seeing all this heavy melanin pigment, that is your clue that that is the beginning of choroid, because we don't have artifactual separation in this one. This is all choroid, including the tapetum. And then this is the visual retina. So if I drew a line here and I came down and I drew a line right there, that's visual retina. So let's zoom in on the choroid. So suprachoroid layer, we can't really see. It's just the junction between the sclera and the choroid. This is all the vascular layer, and you can see there are some fairly, this is artifactual fold right here, but you can see there are some fairly large vessels in this carnivore. This is veins and arteries. And then here we have this strange thing right here, and this is a cellular tapetum. There are two reasons why I know it's a tapetum. One is, well, it's something extra right there between the vascular layer and the choriocapillary layer. So here's my choriocapillary layer. Here's my visual retina. Here is my vascular layer, and there's just something there that wasn't in the pic. And then I can see kind of rectangular shapes. And that tells me these rectangular shapes are like bricks in a building. I have an older house that was built in 1965, and it's got bricks on the outside, and they're really long bricks. They're not your typical size. They're not standard. They're larger, longer bricks. And this reminds me of the brickwork on the outside of my house. So this is called a cellular tapetum, where you can see these bricks. Again, the choriocapillary layer. Now, the retinal pigmented epithelium, or retinal pigment epithelium, is not pigmented right here. That's another clue that I'm looking at the tapetum. Because if the RPE, the retinal pigment epithelium, had melanin in it right here, then the photons of light couldn't pass through those cells and penetrate the tapetum, or bounce off that tapetum, if you will, and get a second chance by passing back through the photoreceptor cell layer. So a photon of light would be coming like this. And if it didn't excite a photoreceptor cell right here in this layer, then it would pass through the pigment epithelium, pass through the choriocapillary layer, hit that tapetum and bounce off it like a trampoline, and be reflected back and get a second shot right here at activating those photoreceptor pigments. So that is what the tapetum is for. Not all species have a tapetum. Pigs don't, humans don't. That's why when someone takes a picture of us with a flash, we get what we call red eye. And that's because we don't have a tapetum. So that flash is making our choriocapillary bed glow. You can actually see the blood, the red like blood in that choriocapillary layer. Kind of creepy and in time for Halloween, right? All right, so there's one more type of tapetum, and that is in herbivores. In herbivores, they have a fibrous tapetum. Okay, so let's zoom in on it. Rotate this around a little bit. Come on, turn for me. There we go. Here, our visual retina is just artifactually separated. It loves to do that because the vitreous is not there. The vitreous body is not there to mash it down. Okay, so what do we have going on here? Well, it's an H&E. That's probably a little terrifying. So here we have the dense irregular connective tissue of the sclera. Here we have artifactual separation between the sclera and the choroid. So this is our suprachoroid layer, where we get that artifactual separation. That's just the junction between the two layers. Here we have the vascular layer of the choroid. It stops about right here. So from here to about here, that's our vascular layer. Here we have a nice large muscular artery. Here we have a large vein. And then what is this? That's weird. It looks like dense irregular connective tissue, but it's wider, thicker, flatter. This is a fibrous tapetum. So here are the collagen layers of the vascular layer. Here's a lot of melanin pigment here. But here we don't have that melanin pigment as much, and we just got these really wide, flat sheets of collagen. This is a fibrous, hence collagen fibers. So not cellular, not bricks, but collagen fibers. That is a fibrous tapetum. Here we have artifactual separation. Look, this is way cool. This is the capillary bed right here. Stretched down here, artifactually pulled away from the RPE. Kind of sounds like RBG, right? The RPE is the retinal pigment epithelium, and here we have very little pigment. There's a little bit here, 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 but not much. That is because the retinal pigment epithelium got rid of its pigment where it overlies the tapetum. So once again, a photon of light traveling this way will come down through all the layers of the retina, and it will try to excite this outer segment of the photoreceptor cells. If that doesn't happen, it'll pass through that retinal pigment epithelium, pass through the capillary bed, hit the tapetum, bounce off of it like a trampoline. Photon of light is coming back, going to penetrate through and try to excite one of these photoreceptor cells. Okay, so that is the purpose of the tapetum. And it's especially present in animals that have good night vision. So they'll have that tapetum and they'll have more rods than cones, and they'll have better.
There you go, Ghostbusters.
Herbivores are surprisingly have surprisingly good vision at night. Sometimes when I go let my cows out early in the morning into another field, as I'm leaving to work, it's still dark right now. And they'll move on out of the woods and into that field, and I'm like, why are you doing that? I can't see. Put out a flashlight. They can see. They're fine. All right, so what's next in our list? Okay, we just finished up the choroid. Now we need to look at the continuation of the uvea, because remember the choroid was part of the uvea. So now we need to look at the ciliary body. And this slide happens to have a good ciliary body. So let me cruise on around here. Now, the ciliary body starts at the ora serrata. And I mentioned that yesterday. The ora serrata is where these 10 or 11 layers of the retina cone down, taper down, and then become just the bilayered epithelium of the non-visual portion of the retina. The retina has the visual portion and the non-visual portion. So this is the pars optica and the pars ceca, the non-visual. So we go from 10 or 11 layers to 2 layers. And when you hit that bilayered epithelium, it overlies the continuation of the choroid, which is the ciliary body, as you're moving anteriorly. The ciliary body is part of the uvea, as is the iris. And it's overlaid with the non-visual portion. Let me turn my email off. It's overlaid with the non-visual portion of the retina. So even though that belongs to the neuroepithelial tunic, because it's so intimately associated with the ciliary body and with the iris, we're going to talk about it at the same time. So here in this ciliary body, we have mostly just collagen and blood vessels, which is kind of what the uvea is all about. But then we start getting smooth muscle. Okay. So you see all these smooth muscle fibers right here. Here's your collagen, which is staining pink with this H&E stain. But then here we have smooth muscle fibers. That is the ciliary muscle that is in the ciliary body. So this is a lot of ciliary muscle. So this animal can accommodate really well, which means it can see far. Okay, and well, up close too. We'll talk about accommodation in just a minute. All right, so this is ciliary muscle. This is the ciliary body. The ciliary body has the flat part, or the pars plana, and then the folded part, the pars plica. Plica means folded. Now the folded part is called the ciliary processes, and they are just an extension off of that ciliary body. What is this for? What's the deal with the ciliary body and the ciliary processes? Well, they form a ring around the lens. So here is a lens, an intact lens with a capsule on it. And here are the ciliary processes. So this is the pars plica, the folded part of the ciliary body. And you can see they form a 360 ring around that lens. And then the ciliary body will be here at the base, all the way outside of this ring-like formation of processes. Okay, so why do we have that? Well, we got to support the lens. And not only do we support the lens, but we control the lens. And how do we do that? Oh, and then probably the most major part is production of aqueous humor. So that's actually quite a lot of stuff going on. So let's take a closer look at that. The ciliary processes at their base give rise to the zonular fibers. So, and these are elastic fibers. So you can see these elastic fibers, they're kind of originating here along this basement membrane on the outside of the inner non-pigmented layer. So this bilayered epithelium right here covering the ciliary body and the ciliary processes, the inner layer that's on the inside of the eye is non-pigmented, and the outer layer that's towards the sclera or more towards the outside of the eye, that's pigmented. The two cell apices meet each other. So the pigmented layer's basement membrane is here, and the non-pigmented layer's basement membrane is here. And that's giving rise to these elastic fibers right here. And all those elastic fibers, or we call them zonular fibers, they come over here and they attach to the capsule, which is a big, thick, modified basement membrane. So we've got all these elastic rubber bands running from that capsule of the lens all the way down between those ciliary processes to the ciliary body. And that happens all the way around. So we're keeping this constant tension. We're pulling on the capsule of this lens, which if you're young like you are, your lens is soft and pliable. So by these ciliary muscles staying relaxed and flattened, and they're oriented what we call meridional or along the equator of the eye, then by staying flat, there's this constant, they keep the ciliary body flat, and so there's this constant tension on these rubber bands, these zonular fibers. So there's this constant tension on the capsule. So we're stretching out that capsule all the way around, and that causes this middle part to stay flat because we're pulling all around the edge. And when it flattens, you can see far away, you're far-sighted. And that is the default mode because that is when the ciliary muscle is relaxed. But if the ciliary muscle contracts, where's our muscle? Come here, muscle. Here it is. If this ciliary muscle right here contracts, it will mound up because it's in this meridional arrangement. So if all these fibers contract, they'll kind of thicken. They'll kind of round up or form a hump here in the ciliary body, the flat part of the ciliary body, the pars plana. Well, what happens? It's not flat anymore. It's rounded up. It bulges and moves these ciliary processes and those zonular fibers just a little closer into that lens. And that means that we're not pulling on that capsule, and that lets this middle part of the lens round up, okay? When that rounds up, then we can see close, we can read things. And that's called accommodation. That helps us focus on things that are close up, but the default is to keep this lens flat and focus on things that are far away. And I think that makes sense. I like to think of it as if you're a species that, saber-toothed lion would like to eat, you want to see that saber-toothed lion from a long way off, right? So you want your default to be farsighted. And that's the way we're made. And that is why when you're older, like me and Dr. K, and our lens fibers have gotten stiff, then when our zonular fibers pull on our capsule, our lens fibers, they don't flatten right here anymore. So then we have to start, going to Kroger or the dollar store and buying our $2, magnifiers so that we can accommodate, so that we can see up close because our lens is too stiff anymore and it won't flatten, it won't accommodate. And that happens when you're in your 40s. So just you wait, something to look forward to. Okay, so we looked at the ciliary body, the ciliary processes. Now, these ciliary processes, one of the main things they do is to produce the aqueous humor. So you can see these blood vessels are going up inside these processes, and that's going to get a real good close apposition with the ciliary epithelium. And these capillary beds have a specialized endothelium with a discontinuous basement membrane, so they're leaky. And they're going to leak a modified fluid, modified plasma, and these epithelial cells are going to transport that and secrete that out into the posterior chamber. So that is how we produce aqueous humor. It's a modification of the blood plasma. So that's something major that the ciliary epithelium does on the ciliary processes. Now that ciliary muscle, it's under parasympathetic innervation. So it'll contract with for accommodation under parasympathetic control. All right, last but not least is the iris. And this is a pretty good iris on this eye, so we'll stay here. So let me rotate it a little bit. So remember that the stroma of the iris is part of the uvea. So it's just a forward continuation. We've got choroid, then we have ciliary body. That ciliary body, that uvea extends up into the ciliary processes. And then it continues anteriorly and forms the bulk of the iris. The iris, of course, has the pupil, the aperture. So this is the tip of the iris. So the pupil would be right here. It has an anterior limiting membrane. So this is anterior towards the cornea. So the anterior limiting membrane would be right here. Then we have iris stroma. Then we have the posterior epithelium. So let's take a look at that. We'll zoom in. And this is, notice this is not called an anterior epithelium, but it's called an anterior limiting membrane. It's just specialized stromal fibroblasts. And it's not an epithelium. It's just fibroblasts that have kind of sealed off this anterior portion of the stroma. And here we have a lot of melanin pigment. So the color of your eyes is determined by the amount of pigment in your iris. So you have blue eyes, you don't have very much pigment. You got brown eyes, you've got the maximum pigment. And everything else is sort of in between. Hazel or green is a kind of a mix in between. Let's see, anterior limiting layer, stroma. There's a lot of smooth muscle fibers throughout the stroma, which is mostly just kind of a dense irregular connective tissue. There's large blood vessels because we're in the uvea. There are gangbusters of nerve fascicles, and they're unmyelinated. So here we have nerve fascicles. So this is an exquisitely sensitive tissue because of all these, all the heavy innervation. And that is why anterior uveitis is so gosh darn painful. Anterior uveitis is inflammation of the uvea of the iris and the uvea of the ciliary body and ciliary processes. And because these things are moving all the time, that makes it even more painful. So you're going to want to, you're not going to want to look at bright light because it's going to be very painful if your iris dilates and you've got a lot of inflammation in here. Posterior uveitis is inflammation of the choroid. So the anterior portion of the uvea is the iris stroma and the stroma of the ciliary body, and then the posterior uvea is the choroid. It's really cool that you'll get collagen fibers, really dense bundles of collagen fibers encircling the blood vessels, so that when the iris dilates or you pull the curtain back on the window, you don't kink these blood vessels, you don't cut off blood flow. Because this is so highly vascular, if you do surgery on the iris, it's going to bleed a lot. Okay. Now we have some epithelium and we have some dilator and sphincter muscles that we need to look at in the iris. The sphincter muscles, all right, are arranged circularly. So if you have a transverse section, you would expect to see those circularly arranged fibers in cross section. And you would expect the dilator muscles, which are arranged radially, you would expect them to appear in longitudinal section if you made a sagittal section of the entire eye. Now you see how when the sphincter pupillary muscles, they contract, they're going to decrease the diameter of the pupil. So here we have fully dilated. So the sphincter muscle is fully relaxed and the dilator muscle is fully contracted. So this is average pupil diameter. This is dilated and this is constricted. So to dilate, the dilator muscles are going to pull back the curtain and you get an increase in the diameter of the pupil. And then when you constrict, you get a decrease in the diameter of the pupil, and the pupillary constrictor muscle contracts and the dilator muscles relax. So what do those look like on histological section? Well, these sphincter muscles you will expect to be in cross-sectional profile, and here they are, smooth muscle, so they've got centrally placed nuclei, and they're going to be just near the apex, the tip of the pupil. So right here, this is your sphincter muscle. This is what draws the curtain closed. Your dilator muscle is kind of funky. It's arranged longitudinally here, but it's part of a myoepithelium. So what do we mean by that? It's an epithelium that became modified and took on contractile properties. So it's not entirely a muscle and it's not entirely an epithelium. Here on the ciliary processes, you can see our two cell layers, our inner non-pigmented and our outer pigmented, okay? On the iris, which is the continuation of that bilayered epithelium of the non-visual portion of the retina, both layers became pigmented. So we no longer have inner and outer differences. They both are pigmented. They look the same. Now we're on the iris, so we're not going to call them inner and outer anymore. We're going to call the two layers anterior and posterior. And that anterior layer, which was the outer layer on the ciliary process, it becomes modified and it has contractile fibers in it. Okay, so this myoepithelium, it's an epithelium with contractile fibers in it. That is the dilator muscle, okay? Dilator pupillary muscle. So we'll zoom out. Here is the sphincter pupillary muscle, and here is the dilator pupillary muscle, right along here, okay? And it's kind of hard to see it because it's overshadowed by that pigment of the anterior, the posterior epithelium of the iris. Okay, I think that gets us through the iris. We've mentioned the iridocorneal angle already, which would be the angle between the iris and the cornea. Now we're obliqued here, so it's hard to tell where the iris ends and the ciliary body begins, but this would be that angle right here. All right, so that takes us to the neuroepithelial tunic. I hope I haven't lost anybody yet. We got to keep chugging. While we're in the area, let's look at the lens real quick. We'll knock it out. We've got that modified basement membrane, that lens capsule. We've got immature lens fibers that haven't gotten rid of their nucleus yet. We have the lens epithelium, which is only under the anterior portion of the lens capsule. So this is the epithelium that will divide and give rise to the lens fibers. Here are the hexagonally shaped lens fibers in cross section, and they interdigitate with one another like Velcro. So these artifactual spaces are between the individual lens fibers. And these are the mature lens fibers that have gotten rid of their nuclei and all their other organelles. And then these are the immature ones that were made recently, and they haven't fully matured, so they still have their nuclei. And here you can see a really cool pattern where we've got some way young lens fibers being formed right here. In the middle, sometimes it'll be just kind of gelatinous in the middle of the lens, and that'll be just a leftover remnant of the embryonic lens, which is just made out of gelatin. And here we can see a longitudinal profile of the lens fibers, and you can kind of see the little Velcro pieces in between those fibers. So they're not really living after a fashion, kind of like the keratinocytes of the stratified squamous epithelium. That stratum corneum is dead cells. These lens fibers are pretty much dead cells after they get rid of all their organelles. Not entirely dead, kind of like a red blood cell. Kicks out all its organelles, its nucleus. It's still sort of living, like a zombie. How about that? Lenses can be zombies. It's a good Halloween thing. All right, the neuroepithelial tunic. Does this have a good one? I think it does. Now, the neuroepithelial tunic, of course, is where vision is. It's all about. It's where it's happening. And there are a bunch of layers in here, and we need you to learn those layers, okay? You need to learn those layers so that you'll understand them when you take your ophthalmology course. So we'll start back here with our epithelium, which is not pigmented because it's overlying a tapetum. So this is our retinal pigment epithelium, and we call it pigment epithelium, whether it's pigmented or not. So this has got a little bit of pigment here and here, but not a lot. These pigmented epithelial cells nourish the photoreceptor cells. They get their nutrients from the choriocapillary bed. They also have the hard job of phagocytizing all the discs that are shed in the morning at first light every day. And they also have cytoplasmic processes that go up and around and kind of protect the outer photoreceptor cell layer. And in bright light, when you're outside, maybe looking at a white sheet of paper, they'll move that melanin up around those photoreceptors to kind of protect them from fatigue, from too many light photons. And then you know how you go inside after you've been outside in bright light and looking at a white sheet of paper and you can't see anything. It all looks kind of dark until your eyes adjust. Well, that's your RPEs here frantically pulling that melanin pigment back down into the basal compartment of the cell and away from the outer segment of the photoreceptor cells so that they can become more sensitive to light photons, which there won't be as many when you go inside a building. The next layer is the photoreceptor layer, and that's belongs to the rods and cones. It's the outer portion of the rods and cones. Rods and cones actually extend from here all the way up to your outer plexiform layer. So from here all the way down here are their nuclei of the rods and cones, and then all the way down to the RPE, the retinal pigment epithelium. That's the full extent of the photoreceptor cells. These are the first neurons in the visual pathway. How do you tell the difference between rods and cones? Well, in some sections you can tell and some you can't. We're lucky here we can tell. So this conical portion and the inner segment of the photoreceptor layer, these are cones. And then all these little stringy, rod-like things right here, and these are the rods, okay? So cone, lots of rods, cluster of rods. Cone, cluster of rods. Cone, cluster of rods. I think you get the idea, okay? Rods we use in low light or night vision, and cones are for color vision and for greater visual acuity. So the sharpness of vision is attributed to cone activity. Next, we have an external limiting membrane, which is right here. We also have an internal limiting membrane. It's kind of like a basement membrane. Here's the internal limiting membrane. Why do we have that and who makes it? Oh, I'm glad you asked. So it's made by the radial glial cells that are a type of astrocyte. So here's where they form the internal limiting membrane, external limiting membrane right here. And then here's where they form the internal limiting membrane. So they're a type of astrocyte, so they're performing a support function. You can see they've got massive cytoplasmic processes and they do a heck of a lot. They're kind of like sustentacular cells, but they're a glial cell, they're a neuronal support cell, okay? So here's your first neuron in your visual pathway, your cones and your rods, your second neuron in your visual pathway, which is the bipolar cell, and then your third neuron in the visual pathway, which is your ganglion cell. And protecting them and nourishing them and giving them support all throughout all those layers and forming those limiting membranes is your radial glial cell, or also known as your Muller cell. So that's M for Muller cell. Okay. Whew, where were we? Retinal pigment epithelium, photoreceptor cell layer, outer segment, inner segment, outer limiting membrane. Then this is the outer nuclear layer, which is just the nuclei that belongs to these photoreceptor cells. So that's rod and cone nuclei. We don't have to know the difference. Here we have our outer plexiform layer, and that's where the rods and cones are synapsing with the second neuron in the visual pathway. So that's right. Let's see, that synaptic layer will be right here. Right here. So here's our second neuron in the visual pathway. And the second neuron in the visual pathway extends between the inner plexiform layer and the outer plexiform layer. Its nucleus is here in the inner nuclear layer, but there are other nuclei in the inner nuclear layer. There are interneurons, which would be horizontal cells and amacrine cells. You don't have to distinguish the nuclei and tell us which ones are amacrine cells or horizontal cells, but you do need to know that there are interneurons that give support to these bipolar neurons and synapse with ganglion cells and synapse with photoreceptor cells. And why do we have those interneurons? Oh, it's complicated, but it's for crosstalk. It's so that all three neurons in the visual pathway can chat with each other and they all kind of know what's going on. They kind of make group decisions on what information they're going to send to the brain. So interneuronal cell nuclei, the radial glial cells, which make the limiting membranes, their nuclei are in this inner nuclear layer. And then the nuclei of the bipolar neurons, the second neuron in the visual pathway. And they run from this synaptic layer to this synaptic layer. Next is the ganglion cell layer. That's the third neuron in the visual pathway. And these are just, they look more like neurons. Okay, so they're multipolar neurons. Here you can see them moving around. Let's see if we can find a nucleus on one of these guys. Yeah, here we go. Here's some nuclei. You can't really tell, but they have euchromatic nuclei with prominent nucleoli. Oh, like this one right here. Here's some good ones. So these are all ganglion cells. These are all multipolar neurons, euchromatic nuclei with prominent nucleoli. These are the third neurons in the visual pathway. They synapse with all these cells that have nuclei in the inner nuclear layer in this really big, wide inner plexiform layer. So the inner plexiform layer is really wide and the outer plexiform layer is really narrow, and these are synaptic zones between the layers of neurons. Those ganglion cells are going to form the nerve fiber layer, and the nerve fiber layer are all the axons of these ganglion cells. And what happens to all of those axons? Well, I'm really glad you asked. So let's take a look here, and I'm going to rotate this. And this is the carnivore eye. And here you can see these axons of the ganglion cells starting to coalesce right here. So here's our ganglion cells. Here's their axons. And this axonal layer is getting thicker and thicker and thicker as we move back towards the posterior portion of the globe. What's going to happen? Boom. Wow. This is going to happen. This is where all these axons of all those ganglion cells, all those multipolar neurons, the their axons come together and form the optic nerve. Now they're unmyelinated right here because we don't need myelin in the way. We still need light to penetrate and get to these photoreceptor cells. So they're unmyelinated, they coalesce, they penetrate through the sclera. Then we don't have a retina right here because it's obliterated by all these axons. They're penetrating through the sclera and an area cribrosa, that means the sieve. And as soon as they penetrate into the sclera, bam, now look, they're myelinated, okay? And they form this really big, thick optic nerve. And that's going to go all the way back to, it's really an extension of the central nervous system. So that is your optic nerve, and it's going back towards the brain, okay? Unmyelinated portion, myelinated portion. Now, where we have all these axons exiting, penetrating, obliterating the retina right here and exiting through the sclera, that's called the optic disc. And this is our blind spot. You can now see why it's blind, because we don't have retina right here. So that's why, if you don't see a car in your rear view mirror, it's because it was right where the image of that car in your rear view mirror was projected onto your optic disc, and you didn't see it, you didn't know it was there. So that's why you got to use side mirror and rear view mirror and turn your head and all of that so that you don't have an accident, because that image was projected onto the optic disc where there is no retina and you couldn't see that car. All right, goodness, we've covered almost everything. There was one more thing I wanted you to see right here. What happens in the posterior compartment? What keeps that retina from detaching? Well, here is an eye cut in two. You can see the lens, and you can see this gelatin all over the lens. That is the vitreous body, and it's a hydrogel. It's well, it's mostly water, but it's hyaluronic acid, making it a gel. And it keeps this white wrinkled membrane right here. That's the visual retina. And you can see the pretty colored tapetum up underneath it. This is the non-tapetal area, and this is the tapetal area. This vitreous body, which is kept hydrated with vitreous humor, it keeps that retina from detaching. That retina isn't really tacked down. So if you rupture your eye, you rupture your cornea, and you know, structures like the iris and the lens protrude out through that rupture, then you've created a lack of pressure here. You created a artifactual space when that lens shifted forward, and then that vitreous body can't keep the retina mashed down, and then the retina will float off. It will detach. So retinal detachment is an emergency. We've got to get everything put back together and get that vitreous body to kind of smash that retina back down, and hope that it will kind of settle back down and get the wrinkles out, and hopefully some of those neurons will still be intact. All right, there was one more slide that you need to look at, and I know you're probably tired, but we do need to look at it. It's the third eyelid. And you might think, well, why bother? Well, I'm having to take online continuing ed courses this month because, you know, we can't meet for our normal continuing ed weekends. And one of my courses that I'm taking, one entire lecture, one entire 60-minute lecture was on the third eyelid. So clinically, it's an important structure. So you need to know the basics of it, because you're probably going to hear about it a lot in your ophthalmology course. It's a fold of conjunctiva, and it's called so many names. The third eyelid, the palpebra tertia, tertia meaning third, eyelid is a palpebra. The nictitating membrane, the haw. There's some other things that they listed that I'd never heard of the haw. But it's important in domestic species. Humans, we've kind of lost our nictitating membrane, and that's too bad. But the domestic species have it, and you'll learn how to retropulse the globe in physical diagnosis and make the nictitating membrane protrude. It has a surface that faces the palpebra, the eyelid, so that's called the palpebral surface. It has a surface that faces the bulb of the globe of the eye, and so that's called the bulbar surface. And the bulbar surface is usually where you have this lymphoid tissue, both diffuse and nodular lymphoid tissue. It is made up of dense collagen. It has smooth muscle in it that is under sympathetic control to maintain tone in the third eyelid. And you'll learn more about the sympathetic innervation of smooth muscle in the periorbita, the little stocking net that surrounds the globe and all the adnexa. It has a supportive framework made out of cartilage, usually hyaline, but can be elastic, but again, usually hyaline, and that's usually T-shaped, that cartilaginous framework. So here is a model. Here's a section of the third eyelid. You can see the lymphoid tissue on the bulbar surface. There's the palpebral surface. And it looks like a shark tail, that hyaline cartilage, but when you make a sagittal section through it, you don't get to see the cute little tails, okay? So there's your shark fin. There's the body of the shark. The body of the shark is this gland. And then there's the skeleton of the shark, the cartilaginous skeleton, which is pretty relevant since sharks have cartilaginous skeleton. So that's the windshield wiper. That's the nictitating membrane. It nictitates or moves when that globe is retracted or pulled back into the orbit. That passively squishes this third eyelid out. It's not an active movement. Last but not least, and actually incredibly important, is this gland. So cherry eye is when you have prolapse of the third eyelid and it sticks out all the time. It's usually red and angry and looks like a little cherry, and it alarms and frightens your clients, and they come running to you and say, help, help, there's a cherry in my dog's eye. And we used to just cut it out long ago. Well, just get rid of the thing. Who needs it? Well, we found out that we do need it because of this gland right here. And this is the superficial gland of the third eyelid, because in larger species, there's also a deeper one called the, well, deep gland of the third eyelid. In carnivores, there's just the superficial gland. It is a compound gland, meaning it has lots of ducts, and those ducts branch to get to all these individual lobules. It's tubulo-alveolar, branching tubulo-alveolar, meaning you'll see branches in the adenomers. This is a duct right above the adenomere. My internet connection is unstable. Okay, let's see. Here's branching. So they're branching, the adenomere branches. So compound means the ducts branch, and then branching means the adenomers branch. Okay, and we can see tubular portions and alveolar portions where we have dilated lumens. There's a little, you can kind of see branching going that way and kind of see branching go that way. Okay, now what does it secrete? It's both a serous and a mucus product. And this one's pretty easy because here you can see the mucin granules. And then here you can see the serous portion. So a watery mucus, seromucus secretion. So compound branching, tubulo-alveolar, some of it's even tubulo-acinar, and seromucus secretion. Okay. So that is the superficial gland of the third eyelid. Don't cut it out. It's incredibly important for keeping the eye moist. If you cut it out, your dog will develop dry eye, or KCS, keratoconjunctivitis sicca, sicca meaning dry. And you don't want that to happen. So now we just tack them back down and we don't cut them out. Third eyelid. So we'll feature that on exams too, so that you'll be familiar with it when you're in your ophthalmology course. And oh my God, Jeffrey, you just scared the mud out of me.
Pretty good.
I'm a Lord of the Rings dwarf.
Y'all are great today. Keep it up. Keep the
Dwarvish.
fun up.